Okay, now coming to the second part of the experiment. So we have prepared the series of standard solutions. Now using these standard solutions, we have to note down the absorbance value of the solution. Okay, now we have to fix the filter to 620 nanometers. So here already I have fixed the filter to 620 nanometers. That means I am passing the light radiation, that is called monochromatic light, having a wavelength of 620 nanometers. Okay, now I am taking one small test tube which is called as cuvette. Okay, first we will be taking the blank solution, which we have to shake it well before we are taking, and first we have to rinse this small test tube and take the solution which is called blank solution into the small test tube, and take a filter paper and wipe off the small test tube and keep this small test tube in the place which is provided here. Okay, this blank solution is for the calibration of the instrument. So using the blank solution, you have to set the absorbance value to 0.00. By turning this knob, you have to adjust the absorbance value to 0.00 because we are using ammonia in this and water. So to nullify that, we have adjusted the instrument to 0.00 using the set absorbance or OD knob. So once if the instrument is adjusted to 0.00, don't touch any of these knobs. Just you take out the cuvette and throw this blank solution, and now you take the 5 ml standard flask solution, 5 ml standard flask solution, rinse this small test tube and again take the solution into it fully. Take 5 ml solution, and before you are keeping, please remember that you have to wipe off, clean this on all sides and put it in the place and note down the absorbance value. It is showing as 0.11. You have to note down here the absorbance value for blank is 0.00 and for 5 ml we have got 0.11. Okay, now you have to take out the cuvette. I am not touching any of these knobs. Throwing the solution. Now I am taking 10 ml standard flask and taking a small amount of the solution and rinsing this, and again this solution what we have prepared into this, and before again I am cleaning this cuvette with a tissue paper. It should be clean. Then I am keeping it in place, putting the lid. Now I am noted down the absorbance value 0.22. 0.22. Okay. Now again I am taking out this cuvette. Now I am taking 15 ml labeled standard flask. Again rinsing the cuvette and taking the solution into this cuvette. Cleaning on outside. Putting it back here and noting down the absorbance value 0.31. So what I am observing is, with increasing the concentration of copper sulfate, the absorbance value is increasing. So Beer-Lambert's law states that absorbance is proportional to concentration. Okay. Now I am taking 20 labeled standard flask. Rinsing the cuvette, taking the solution into it, cleaning this, putting it back and closing the lid and noting down it is 0.39. Now I am taking the last standard solution, that is 25 ml standard flask solution. Rinsing the cuvette again with the solution and taking the solution. No need to wash the cuvette with water because we are increasing the concentration, so it will not affect the readings. So only with the present solution you have to rinse before you are taking that. And noting down the value, it is 0.47. Now we have taken out the readings of all the solutions except the test solution. Okay, before we are taking the test solution, rinse it. We don't know how much amount of copper sulfate was given here. We have to find out. So now you have to rinse this with 2-3 times because we don't know the concentration of this. So better rinse this cuvette 2-3 times and take the test solution into the cuvette and clean this using the filter paper. And keep this here and note down the absorbance value of the test solution. It is 0.29. Okay, 0.29. So I have noted down for all these solutions the absorbance value. So okay, what I have observed, when I am noting down the absorbance value, according to Beer-Lambert's law, when I am increasing the concentration of copper sulfate, the absorbance value also increases, and I have noted down the values in the tabular column. And now using these values, we have to find out the amount of copper present in the given copper sulfate solution.